I'm Daniel Levine, and this is the Bio Report. Earlier this year, Profluent announced it had edited the human genome with Open CRISPR-1, the world's first AI-created and open-source gene editor. Though the open-source aspect helped garner attention for Profluent, it also served as a demonstration of the company's generative AI platform to create novel CRISPR gene editors. We spoke to Hilary Eaton, Chief Business Officer for Profluent, about how the company's generative AI engine works, its business model, and why its platform has the potential to create other protein therapeutics of varying modalities. Hilary, thanks for joining us.
It's a pleasure to be here. Thanks for the invite.
We're going to talk about AI, Profluent, and its efforts to use generative AI to read and write any class of proteins. Profluent's initial focus is on gene editing. Why start there?
So at Profluent, our first and foremost ambition is to solve problems that maximize impact on society. Gene editing is a relatively new modality, but from the very beginning, folks had recognized its enormous potential to have an outsized impact on human lives by creating lasting cures to disease. So essentially, instead of trying to fix symptoms of disease by taking a pill every day, why not go to the root cause and fix broken DNA with a one-and-done treatment? And so the impact factor is really aligned with our goals. Now, what's blocking us from getting there to this new era of genomic medicine, of fixing broken DNA? First, it's really that we've mostly been limited to finding things in nature, to serendipitous discoveries. So an interesting anecdote here is that some of the initial work done with CRISPR-Cas enzymes happened at a yogurt factory, where nobody was trying to develop medicines, they were just trying to make the yogurt manufacturing process better. And so at Profluent, we view AI as the path forward for custom solutions and control. So moving from a world of accidental discovery to more one of purposeful and precision design. And gene editing is really a perfect match because the systems there are so incredibly complex and intricate that they really need and deserve AI to bring them to the next level.
Well, what are the limitations of existing CRISPR enzymes for gene editing?
So I would start by acknowledging all of the incredible progress that's been made by pioneers in the field of gene editing over the last decade. Seeing CRISPR medicines like Casgevy for sickle cell disease getting approved is just a phenomenal advance for patients. So where did all of these systems originally come from? As I alluded to, they were essentially copied and pasted from other organisms that exist in nature. Now, an enzyme that evolved in nature to do a certain thing may not be a perfect match for every therapeutic use case we'd like to apply it to in humans. What do I mean by that? If you pick a particular disease with a particular DNA mutation that's prevalent in patients or a group of lots of different mutations, it's very unlikely that there's a natural enzyme that just randomly evolved to have the perfect PAM, the perfect efficiency, the perfect size, the perfect stability, the perfect off-target profile, et cetera, et cetera.
What's the potential, though, to use generative AI to create new CRISPR enzymes rather than relying on wild-type enzymes? And there are a number of companies that are mining the bacterial world right now to find a broader array of CRISPR enzymes.
That's a great question. So say our starting material is an enzyme from nature, and we want to use it for something slightly different. We want to adjust its function. So how do we go about doing this? Because protein engineering is not a new thing, right? We could carry out manual protein engineering techniques like random mutagenesis or directed evolution. These are pretty labor intensive and time consuming, even with some of the high throughput advances that have been made. And often they aren't hypothesis driven, which means that what you typically get after months or years of effort is just really incremental improvements at best. So next up are companies that do metagenomic mining. essentially searching a wider array or diversity of organisms for similar sequences in the hopes that one of them is going to match the altered criteria they're looking for. But this is a little bit of a needle in a haystack approach, and it often results in functional trade-offs. So, for example, maybe you find a smaller enzyme that fits into your desired delivery vehicle, but you take a cut on other attributes like efficiency or binding specificity. So you often see these systems where essentially you find something that matches what you need in one parameter of the function of the protein, but you take sacrifices in these other areas. And so if we fast forward to what Profluent is doing with next generation LLMs, large language models, we don't have to be limited by natural sequences, by manual labor, or by functional trade-offs. You know, the combinatorial space of a single protein is greater than the number of atoms in the world. And there's no way that there's any manual technique that can work through that many potential combinations, but generative AI can parse all of those possibilities in a matter of hours. And so I guess, you know, to summarize, gene editing systems are so very complex, and there are so many knobs and properties that you could optimize and maintain. And that's precisely why we need to move towards a future of intentional design versus random discovery. AI is sort of uniquely capable of multi-attribute optimization.
Profluent garnered some attention when it announced that it had released the world's first open source AI generated CRISPR gene editor and that it had successfully demonstrated precision editing of the human genome with it. Why release this as an open source editor?
That is the most frequent question we have been getting lately. So, a few points here. A lot of gene editing companies have significantly overlapping pipelines. Now, that's partly because it's challenging to deliver gene editing medicines to certain tissues in the body, but it's also partly because there are very few genetic diseases that have large patient populations. At Profluent, we really want to democratize gene editing and spur innovation for many use cases because there are hundreds of other rare genetic diseases that could benefit from this technology. And this is really an emerging theme in the field. Think of the recent Beacon initiative between Danaher and Jennifer Doudna's Innovative Genomics Institute. It's all aimed at sort of getting this technology out to more patients, no matter the size of the patient population. And sort of beyond sort of the trends in the field and Profluent's ambitions, it's actually also a personal passion of mine. I'm a rare disease patient and the mom of two little girls with the same rare disease. And so it's just personally so gratifying to see all of these new initiatives emerging that will get more medicines made for more patients. And so we made the decision to release OpenCRISPR open source. And it just felt so great to get it out to the community to test and further build upon. I'd also, make the point that OpenCRISPR-1 is essentially just a byproduct of what we can do with our platform. We can build so much more along two different axes. The first is breadth. So gene editing is where we started, but we can also go into many other verticals, other types of proteins, antibodies, etc. There's also a lot of depth here in terms of within a given vertical, really, no matter which modality you're looking at within gene editing, we're able to create very custom bespoke solutions. And so since our vision is much more than just open CRISPR and simple nucleases, it wasn't very worrisome to us to open source it freely because we have this portfolio of millions of CRISPR analogs and none of them are static assets. So what do I mean by that? Historically in the field, you'll see sort of these buckets of IP or ownership where there's, some version of a CRISPR-Cas enzyme that is owned by a single entity. And you often don't see a ton of iteration, or maybe you see some sort of incremental tinkering. And then they, create therapies and they license them out to big pharma, et cetera, et cetera. All of the sequences that are in our portfolio, they don't need to stay identical to what they are now. The more data that we get from wet lab validation, either positive data or negative data, that can be used to fine tune our AI models to make sure that the next version that we release is even better.
I should note that while you have made OpenCRISPR available for free, you did file patents for it. Why is that?
Yes, so we did file patents on OpenCRISPR-1 and many other sequences in our portfolio. And essentially, our aim was to be the first to file on this so that we could ensure we could dedicate it to the good of the public and choose to license it for free. And while we're committed to making this IP available to the community at no cost, we also want to do so in a safe and ethical way. So our open source IP license has certain excluded uses, like not being able to use OpenCRISPR-1 for human germline editing, for example.
What's been the response of people indicated interest in making use of it?
So the response, frankly, has been overwhelming. It has surpassed our wildest dreams. Just going, through some of the stats, millions have seen our work. Tens of thousands of folks have accessed the sequence on GitHub. And within 24 hours, hundreds of people had applied for access to our free license. Really, practically any institution under the sun, from academia to industry. And this really builds upon the incredible adoption of CRISPR as a whole in life sciences. It's transformed so much in biology, right? And not just human medicine, but agriculture, a wide variety of other fields. And so we really wanted to just introduce a new jolt of energy to continue pushing this field forward. From my perspective as not an AI expert, but a biologist turned business development, it feels like we're on the cusp of a major inflection moment. There's lots of hype about AI out there. But I would say that my experience over the past decade has been more AI as it might be used to analyze clinical trial data or to improve the efficiency of manufacturing processes or data analysis. And I think what we're building towards here, and what OpenCRISPR is a great example of, is the power of AI to fundamentally transform the earlier stages of drug discovery and drug development.
I think for people who live in the gene editing world, they might be surprised to learn that Profluent's origins go back to a research project led by Salesforce called ProGen. How did that work lead to the creation of Profluent?
Yes, it is a somewhat unique, unexpected origin story. So it all starts with natural language processing, which is research into how AI can understand how to read and write human written languages like text. And so, harkening back to before there was a ChatGPT on everybody's phone, locked away in a handful of these big tech labs, researchers were witnessing these great advances in AI enabled by models like the Transformer. So, to give a concrete example, you rewind 10 years or so, maybe less, and when you were trying to type a message on your iPhone, it could barely figure out what the next word is and finish your sentence. But then if you fast forward to where we are now, you can go into ChatGPT and it can generate an entire essay in whatever style you want. And that was all enabled by some of these, quantum advances that were made in these big tech labs. And so Ali worked at our founder and CEO, Ali Madani, worked at Salesforce, headed a lot of these generative AI efforts. And having sort of conquered the world of chatbots, he really wanted to use AI to make a positive impact on society and medicine and use it to tackle previously unsolvable problems. And so it occurred to him that you could move from using AI to read and write English language to using AI to read and write the language of biology. Fundamentally, these AI models learn through examples, and nature has provided us with so much data to learn from all of these years of evolution. And so Ali built ProGen at Salesforce and then needed to test the research rigorously. After all, when it started spitting out sequences, he couldn't vet them. They weren't English language, they were protein sequences. And so he collaborated with folks in academia like James Fraser, who's the Vice Dean of Research at UCSF, a protein structure scientist. And I'm sure Jamie was a little bit, intrigued by somebody from Salesforce sending him random protein sequences. But fundamentally, the researchers at Salesforce wanted to ask the question of whether these models were producing functional proteins or not. And from those core academia collaborations, it became clear that yes, indeed, these Gen AA models were working and working really well. So it kind of morphed from being a research idea, a proof of concept, to then a going concern. And Ali wanted to go tackle challenging problems that would benefit society and be uniquely unlocked by AI. And so he left Salesforce to start a company that over the past couple of years has built increasingly more sophisticated foundational models that can read and write therapeutic proteins from scratch. And he got funding from investors who shared this vision for the future, folks who've been in the trenches of biotech, but also tech visionaries like Jeff Dean and Fraser Kelton, who was the head of product for OpenAI and DALL-E and ChatGPT, who really understand what it takes to take some of these research concepts and translate them into commercial success.
What is the language you're using? Is it protein sequences? Is it genetic code, RNA?
So fundamentally, it's protein sequences with a variety of other data layered on top.
What is the data that you've trained these models on? Are there publicly available data sets? Are you generating proprietary data to train these systems?
So it's a combination of both. So essentially, there's a lot of genomic data out there in public data sets, but one of the issues with being able to use it is that often it's disorganized or it's in different formats. So you have to do a lot of heavy curation to get it into a format that's usable by these AI models. So that's kind of like one bucket of the data that we use. There's also proprietary data that we generate ourselves, and we combine all of these buckets, to align for functional design tasks that we're most excited about. So as I was kind of alluding to earlier, part of that is sequence data. So our frontier models currently have over 40 billion protein sequences within their genomic context. But that's only one part of the equation, right? So, it's phenomenal to see a sequence. It's phenomenal to be able to predict how it's going to fold, but none of those things necessarily equate to functional outcome. And that's what we're trying to do, is generate de novo proteins that work the way that we want them to. And so on top of that sequence data, what we've layered into our data sets is protein-protein interaction and other biological data that helps capture the complexity of, where are we sending these proteins? They're not going to be operating in a vacuum. They're going to be interacting with other molecules in the specific tissue that we're sending them to. And so we're really just excited to be able to capture that full complexity of biology to make sure that when we create these novel proteins, they don't just hypothetically function, they actually work when we put them into their target environment.
How does the process work? Are you able to describe characteristics you're seeking in a CRISPR enzyme and have the system generate a list of protein sequences? Does it score them in any way?
Absolutely. We can steer our models with the exact attributes that we desire. So, to continue the ChatGPT analogy, it's similar to providing a prompt there. You say for the use case that I'm looking at, my ideal CRISPR enzyme would have the following attributes. Maybe you care about all of these attributes equally, or maybe some of them are must have and others are sort of nice to have. And the model, chugs through those prompts and then generates libraries of sequences with model confidence scores, because it's no good if your AI model generates millions of sequences and you have to then test all of them. You want it to get really accurate at predicting which are the best, and then you test just the best and a few others and use that to continue to evolve towards the ideal protein sequence.
Profluent does have a wet lab. At what point do you move from AI to the wet lab, and does that data feed back to improve the AI in any way?
It's a great question. So, again, we aren't performing directed evolution. We're using these AI models to generate sequences from scratch. So we're not starting with known wild type sequences and introducing mutations. The models are generating these de novo sequences. Once we have the first generation of sequences, that flips over to our wet lab, where they perform validation, some of the standard assays that you'd have in a gene editing pipeline: on target, off target, immunogenicity, etc. And that creates sort of like a flywheel or a closed loop system where, you've got this really tight coupling. AI model turf stuff over to wet lab generates data that feeds back into the model, and the models learn from both positive and negative data. They're then able to generate children's sequences that are even better than the parent sequences, and thus the sort of generational or generative AI metaphor. And it's just such a rapid process compared to any manual technique that I've seen used in platform groups before.
What's the business model? Do you expect to develop your own pipeline of therapies, license out CRISPR enzymes, or work as a service provider?
So we are very partnership focused. The theory here is play to our strengths. We have unparalleled capabilities to generate bespoke proteins, and we want to partner with the best folks in a given vertical, like gene editing or antibodies. I'd stress AI is not a magic wand. It's not going to solve everything. It needs very tight coupling with folks who intimately know the biology, the manufacturing, the clinic, etc. And I mean, the key here is that we really want to totally flip the drug development paradigm upside down. So rather than start with a protein from nature and try to figure out what it could fix, we want to start at the end with the exact disease problem that our partner is trying to solve and then customize an exact solution. So what we're looking for on the partnership front is really big, meaty problems that are suitably ambitious and complex to necessitate AI to solve them.
So there's obviously a wide range of applications for proteins, a large world of protein therapeutics. Are you focusing exclusively on CRISPR enzymes right now, or are you doing in parallel other protein therapies?
We've already made breakthroughs with our foundational models in other modalities, in antibodies and other enzyme classes, and all I can say is stay tuned for more.
In March, the company raised $35 million. That's on top of a $9 million seed round. How far will existing funding take you, and what's the plan for raising additional capital?
Yep, so we've raised $44 million to date, and that is sufficient funding for plenty of years to come. We're using that funding for continuing to build our next-gen foundational models, to grow our proprietary data set, and to expand our wet lab footprint and validation capabilities. And again, we're really just focused on partnerships, right? The lift on our side in terms of in silico and generating these libraries of sequences and sort of the best hits, that's the value that we bring to these partnerships. And we're looking to partners who share those ambitions and have these, really meaty therapeutic problems that no existing tool in their toolkit, be it gene editing, antibodies, etc., has been able to solve.
Hilary Eaton, Chief Business Officer for Profluent. Hilary, thanks so much for your time today.
Thanks so much.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.